Welcome to Darwin 2009 at the University of Louisville, October 23rd, 2009. Dr. Awdhesh Kalia of the University of Louisville will be making a presentation on evolutionary genomics of pyogenic streptococci.
I'd like to start off by thanking Lee and Martin for organizing this wonderful celebration in honor of Charles Darwin, and also for giving me the opportunity to present some of the ongoing work in my lab. It's going to be a bit of a fall from cancer to bugs that cause strep throat, but I hope you'll bear with me. And I also realize it's getting close to dinner time, so I'm trying to make it as fast as possible. This being said, if you want to stop me in midway and ask a question, please feel free to do that. So I'm just going to talk a little bit about our work in the lab, which is trying to understand the origins of virulence and hypervirulence in two very related pyogenic streptococci. Pyogenic streptococci are those that go on to cause pus, essentially. So just to give you a little bit of a background on the bugs that I'm going to talk about, the major bug that my lab focuses on is Awdhesh streptococcus, which is gram-positive bacteria which grows forming chain. And most of us at some point in life have encountered this pathogen in the form of strep throat, which is the most common outcome of this. Although really not very serious, strep throat alone in the United States is responsible for almost $3 billion in annual losses, as we measure everything in terms of finances. So the bug has two ecological niches where it circulates within in the human body. It's human-specific pathogen, and most commonly it colonizes the throat and the skin. However, depending upon where this bug colonizes, throat or the skin, the clinical consequences of this colonization can be radically different. Most commonly, these are mild and self-limiting. In the case of throat, it can cause pharyngitis. In the case of skin, it can cause impetigo. And both of these clinical symptoms are mild and they're very self-limiting. And most subjects experience these symptoms repeatedly, particularly during adolescence and young age. Repeated attacks of strep pharyngitis and impetigo can, however, have devastating and serious consequences in certain susceptible individuals. For example, repeated pharyngitis can manifest in scarlet fever, which used to be very common in the 1800s, late 1800s and early 1900s, and is unfortunately rising up again today in certain parts of the world. Repeated impetigo can lead to erysipelas and cellulitis, which are basically invasive diseases, invasive infections of the skin. And in certain individuals, such repeated attacks can manifest in permanent damage to the heart, where it can cause rheumatic fever, rheumatic heart disease, permanent damage to the kidney, where you can have post-streptococcal glomerulonephritis. And in most recent times, there's been an emergence of strep strains that cause particularly severe infections. For example, necrotizing fasciitis and toxic shock like syndrome, which have earned this notorious fame in being called as the flesh-eating bug. One of the major things that we are interested in is that given this large spectrum of diseases that can occur as a consequence of strep infection, we don't have a reliable clinical marker that can predict that if an individual is infected by group A strep, which of these diseases is that individual most likely to develop. Now, part of the reason strep causes so many diseases is it's fully equipped with a battery of what we call as virulence factors. It makes several proteins, several bacterial components that contribute directly to the survival of the bacteria in the face of a mounting immune response from the host. And it also secretes a large battery of proteins, some of which are toxins, into the environment, which can go on to cause pathology directly. Some of the important toxins include the pyrogenic exotoxins, and I'm bringing these up specifically because these will feature again later in the talk. These pyrogenic exotoxins, they're called pyrogenic because their mode of action elicits a fever in the host. Okay? And all of these toxins are phage encoded. They're not a part of the strep genome or strep chromosome per se, but they're encoded on various phages that are resident in the streptococcal genome itself. apart from the secretory factor, the other major virulence factor of group A strep is the so-called M protein. Now the M protein is mostly an antiphagocytic protein, but apart from its role in bacterial virulence, it's very useful in terms of epidemiological tracking of streptococci, of group A strep. For example, epidemiological tracking with M protein over several decades, starting from early in the 1900s till today, it still continues. A couple of features have become evident about the epidemiology of strep infection itself. First, different strains of group A strep, each easily distinguishable in terms of the M protein it carries. So you can have group A strep with an M type 1 or an M type 6, as is shown over here. Periodically, different strains of group A strep, they wax and wane through the populations. Okay, so one serotype reaches a peak, then it falls off, and another serotype reaches dominance in the population. Using the same epidemiological marker, what we've also observed is that over large periods of time, okay, since this is one of the oldest pathogens known, so we've had a lot of epidemiological tracking data on this. This pathogen has undergone several cycles where it had a high incidence worldwide in humans, and then it begins to fall even before the introduction of penicillin, stays low after the introduction of penicillin, and since 1980s, for reasons that still remain unclear, this bug has made a comeback, particularly in the United States and in Western Europe. And this re-emergence was dominated mostly by two types of group A strep strains, those that belong to M type 1 and M type 3. In either cases, we do not have a clear idea about why group A strep populations show this kind of a dynamic in natural human hosts. So this is one of our major interests. What are the genetic factors that have contributed to the re-emergence of these group A strep strains? Now, while this was happening in the early and mid-1980s, a lot of people focused on group A strep. And a lot of people, for that reason, missed another bacterium, which is very much similar to group A strep, is human-specific, that also began to show a higher incidence of disease associated with its colonization. Now, these bacteria are called as the group C and G streptococci. Now, the different groups, the group A, C, and G, they basically reflect different sugars which are present on the cell wall of these streptococci. Now, these streptococci, they're considered as commensals, routinely isolated in the clinical laboratory and paid no attention whatsoever. Okay? They look almost identical, entirely identical to group A strep. On a blood outward plate, they show this kind of a beta hemolytic appearance. They lyse red blood cells. And under microscopy, they form a similar kind of chains as is seen with group A strep. The most remarkable, several of the known virulence factors of group A strep, like the M protein, the T antigens, and so on and so forth, most of these have conserved homologs in the genome of group C and G streps. What I mean by conserved homologs is that even the lengths of the open reading frames are preserved. The sequences have just diversified as a consequence of speciation. So, as a postdoc, I got interested, as a PhD student and then as a postdoc, I become interested in asking what would happen since the ecological niche of both group C, G, and group A streptococci is identical. What will happen if there would have to be a chance meeting between these different streptococci? So our work over the last 10 to 12 years, starting from my PhD days, basically can be summarized in this one slide, which basically led us to establish that there is a rich history of interspecies gene exchange between group C and G streptococci and group A streptococci. But there's something strange about this gene exchange. As you will see in this model, you have some big arrows and then you have some small arrows. What is strange about these arrows is that oftentimes these arrows point only in one direction. Okay? There is asymmetricity towards this gene transfer. Genes move from one pathogen to the so-called commensal. So let me explain what I mean exactly by asymmetric horizontal gene transfer. Now, in a typical analysis of phylogenies from homologs, okay, closely related species, both species tend to form their own clusters. Okay? As a consequence of speciation, genes tend to accumulate changes and then diversify. So what you see over here in this red cluster is the species cluster of group C and G streptococci. And what you see over here is the species cluster of group A streptococci. Now, when intragenic recombination occurs between these two homologs, you typically see these intermediates or reserves. Now, this is not asymmetric gene transfer per se. I don't know how clearly you can see it, but these alleles, like the SCA36, the SCA7, and these are alleles which have been found from group A strep populations, which are smack in the middle of the group C and G species cluster. Okay? So this is what we mean by recent asymmetric horizontal gene transfer, where alleles of one clonal frame are often found in the clonal frame of another species, and there is no back movement of such genes. Now, what would be the relevance of this? We hypothesize that the directionality which we see is mostly from group A strep to group C and G strep, and very little gene flow back, mostly in the case of the M protein, from the commensal to the bathymen. So much so that the commensal genomes, if you look at a variety of isolates, they tend to provide a very heterogeneous mosaic-like feature, with some genes which are identical, those in green as is shown over here, to what should be actually the genes of group A strep genomes. Okay? So our two hypotheses that emerged from nearly 10 to 12 years of work was that this kind of strong directionality probably reflects or probably helps the commensal to attain better fitness in its human host, assuming that this earlier divergence occurred in two different hosts, and the commensal has moved only recently into the human host itself. Okay? And in the case of group A strep, we hypothesize that the backflow, the little backflow that happens, particularly in the case of virulence factors like SCA and streptokinase and so on and so forth, probably aids in the emergence of newer phenotypes of group A strep, most possibly the emergence of hypervirulent strains in the mid-1980s. So some of the long-term questions that my lab is interested in addressing are why do genetically related species that share identical ecological niches, that share a battery of virulence factors and that seemingly recombine a lot, that differ extensively in their virulence. Now, even in the mid-80s, there has been some increase in the incidence of GCS-GGS infection, but there is a catch. Most of the subjects tend to be suffering from comorbid immunosuppressive conditions. Okay? And overall, the contribution of GCS-GGS to disease remains much below that of GAS. Our second question is, what is the biological relevance and the extent of asymmetric gene flow that we've seen from limited sequencing-based studies from group A strep donors to group C and G strep? And two long-term hypotheses that we want to test include that differences in the virulence of group A and group C and G streps could possibly reflect species-specific content differed from one species to another, or otherwise, differences in virulence might be a consequence of differential regulation of the orthologs that are so beautifully conserved in both of these species. How those orthologs are expressed and regulated in two different genomic contexts may have an important bearing on what the final phenotype of colonization might be. Now, to address these questions, one of the first steps my lab has taken is to sequence the entire genome of the first group C strep isolate. And the approach that we have taken is fairly standard nowadays, at least. We use the 454 pyrosequencing technology, analyze the sequences for their quality, and then did what needs to be done in order to make sense of the millions of base pairs of DNA sequence data that comes surprisingly nowadays in less than a week. And there's an interesting story because when I started getting the sequence, that was 2006, and I was thinking that it might take some time to get the sequence. The sequence was back in 1 week and I had no clue what to do with it. And I was completely unprepared as to what am I going to do if the genome sequence arrives today. So it took me about quite some time to figure out what to do with that sequence. So this table just summarizes some of the key statistics of where the genome data today stands in comparison to, so this is the group C genome that we have determined, and these are the genomes of some of the reference group A strep strains. Now and we go through some of these features in detail as we would cover that piece of data. Now the key features of the group C strep genome, as you can see from this multiple genome alignment where we have the genome of group C strep aligned with several different N types of group A strep genomes, is that the group C strep genome is somewhat bigger than the group A strep genome in general. It's about 2.1 megabases in size, whereas the typical group A strep genome tends to be about 1.8 to 1.7 megabases in its total content. Now, with this overtly complex slide, there are only two take-home things that I want you to take with you. One is the colors have a meaning. What the colors show is genes which are common between different genomes analyzed. Okay? The heights of these columns reflect the extent of homology between orthologs present in different genomes. And the white spaces that you see liberally interspersed in several genomes reflect regions of the genome which have no homology in any of the genomes present or any of the genomes that are analyzed. Okay? So the natural question is, what is similar, the colored region, and what is species specific, which would be regions which are white and have no homology. But before we went into answering that question, what we wanted to know, since our previous data has suggested this idea of asymmetric horizontal gene transfer, was to ask ourselves, given this level of homology between the two species, what is the global extent of asymmetric gene flow from GAS to group C strep and group G strep? Now, analysis of these genomes revealed that the extent of asymmetric gene flow is rather striking. Nearly 8% of the GCS genome, of the group C strep genome, is identical to group A strep genomes, which is really, really dramatic. It's not somewhat similar, it's identical, by which what I mean is sequence identity greater than 98%. Okay? Because for most species, we accept that species clusters form when the divergence exceeds 2%. So within that, we consider it the same clone frame. So what you see over here is the extent, the number of genes in group A, in group C strep genome, which are completely identical to group A strep. And this number is a little bit outdated because we now have new annotation of the genome. This is an ongoing project and numbers change almost on a daily basis. These genes have conserved orthologs of nearly about 60 to 65% of the genome in group C, between group C and group A genome. And then you have a percent of genome which has no relationship whatsoever with group A strep. Now here I'd also like to make another important point, particularly for those of us who have questioned the efficacy of bacterial speciation in particular because of rampant horizontal gene transfer. If one looks at limited sequence data emanating from multi-locus sequencing projects or limited gene sequencing, one tends to see blurring of species boundaries. But even with this dramatic extent of asymmetric horizontal gene flow, the two species clusters are completely finely preserved. So it looks like that the questions associated with whether bacterial speciation is real or should be used at all seem more of scale than of biology of data analysis. So once we figure this out, that there is nearly 8% of the genome which seems to have been acquired by group C strep from the pathogen, the next question we wanted to obviously know was, so what is so specific about these genes? What kinds of genes are more susceptible to horizontal gene transfer? And this is where our first shock came. Okay? Now one would expect Well, the epidemiology is changing. Group A strep is becoming hypervirulent. Group C and G streps are becoming more virulent. So we should see a movement of virulence genes from the pathogen to the commensal, which would be the most parsimonious explanation to suggest an increasing virulence in the so-called commensal. Well, we did not see any virulence genes which were being transferred from the pathogen to the commensal. Instead, and this is just a pairwise comparison of group C strep genome with the M type 3 genome, which shows that predominant nature of genes that are asymmetrically transferred from the pathogen to the commensal are genes that encode metabolic proteins. Proteins such as dehydrogenases, synthases, kinases, aldolases, and so on and so forth. Okay? Nearly 88 of the 200 or so odd proteins that we now know that share a common origin, that come from the pathogen to the commensal, are metabolic. Another key feature: a large variety of genes tend to be transporters. Okay? These are transporters belonging to the ABC transporter families, the PTS components, sugar transporters, and metal ion transporters, for example. And this is to me indicating that there is a positive move towards group C strep acquiring genes from a well-adapted human pathogen to facilitate its own adaptation to a relatively new host to which it has moved relatively recently. One of the most shocking features of this asymmetric gene movement was the acquisition of genes involved with translation. And historically, genes such as those which are involved in translation, replication, transcription, so-called information genes, they tend to be refractory to horizontal gene movement. Okay? Because they're so well adapted to a particular genome, they seldom seem to move horizontally from one species to another. And that is the basis for why we use 16S ribosomal RNA as a marker for studying different species or identifying different species in complex communities. But in the case of this particular dynamic between the commensal and the pathogen, we see a wholesale movement of genes associated with translational machinery from the pathogen to the commensal. Okay, so the key feature that emerges is that movement of these genes, which are not directly involved in virulence per se, not directly involved in causing pathology per se. Okay, what benefit could they potentially provide to the recipient? So our hypothesis right now is that given the nature of and the makeup of these genes, it seems more likely that group C and G strep have moved recently into the human population and are taking advantage of this asymmetric gene flow to adapt rapidly to their new environment. So that's one possible scenario. Now this scenario changes a little bit when we talk about another class of proteins that seems to have moved asymmetrically from the pathogen to the commensal, and those are the transcriptional regulators. Okay? Now, when we looked at the transcriptional regulation machinery, the complete complement of transcriptional regulators in group C and G strep, we saw a variety of transcriptional regulators, and most of these are not important. Okay? What we saw was that group C strep tends to carry more transcriptional regulators. At an average about 125 transcriptional regulators compared to group A strep, which at an average in various genomes taken together, carry about 75 to 85 transcriptional regulators. Okay, so there is an expansion. Aside from the fact that several of these transcriptional regulators are shared between the two species, there is a relative expansion of transcriptional regulators in the commensal itself. Now you might notice that some of these transcriptional regulators are in red. Okay? Now the most important cases that I'm going to point out are those of the two-component response regulators, called as listed over here as the CSR-R and the CSR-S. Now, the reason I bring this up is because these two-component response regulators play a very important role in the pathogenesis of group A strep infections. Now what these two component response regulators do is they sense environmental signals, okay, and then transmit that information via this sensor kinase system to regulate the expression of several different proteins in group A strep, some of which are key in dictating the response of the pathogen to the host, to the various stimuli presented by the host. Okay? Also, in terms of nutrient adaptation, in terms of various other functions to negotiate with the host immune response, and so on and so forth. Deletion of such two-component response regulators from the pathogen directly abrogates the virulence potential. Okay? Now, one of the key things in terms of acquisition of transcription regulators is that the group C strep seems to have acquired a copy of this two-component transcription regulator from group A strep. Completely identical. It has its own native complement of CSRR and CSRS, and it has on top of that acquired a copy of this two-response regulator from the M-type 3 group A streps. Now, we don't know how that changes the dynamic or behavior or response of the commensal to the host environment, but that's what we think right now is probably the case. Now, given this extent of horizontal gene transfer, okay, so accepting that there is horizontal gene movement, things are moving in one direction from pathogen to the commensal, one question we've been also interested in is what are the vehicles? How does DNA get from one species to another? What is physically moving this DNA from one species to the other? Now, in this case, what you see is a comparison of the whole genomes of the group C strep genome with the M-type 3 GAS genome. And the reason we are focusing more on M-type 3 is because that's the type of group A strep strain that's dominant currently in the US and Canada and is associated with the flesh-eating phenotype. Now, one thing that emerges strikingly from this genome-wide comparison is that if you focus on these areas which are white, pointed here by these arrows, now these indicate sites where bacteriophages are inserted in the genome of group A strep. Okay? And if you compare the homologous region in group C, aside from this difference, the two regions are completely synthetic, completely identical. If you look at this purple patch here, and if you look at it, if you remove this white section, the two regions of the genome are perfectly identical. Okay? And this pattern seems to appear again and again and again. Nearly identical regions of the genome have been pushed apart by insertion of multiple bacteriophages. In the case of group A strep, each of these bacteriophages carries an exotoxin encoded within its genome. Okay? So while this is very nice, we see similar patterns even in the group C strep gene, indicated to you over there in green arrows, which indicate sites of bacteriophage insertion. Now, the fact that these are all in white regions means that they don't share any significant homology with each other. These are all different bacteriophages that are inserted in the two respective genomes. So, seeing this kind of integration pattern, it was easy to imagine that such phage insertions are probably driving the ecological and genetic divergence between the commensal and the pathogen, superimposed upon the fact that otherwise the genome tends to be more or less similar. Okay? Now, this is just an example to indicate to you the phage insertions in the commensal bacteria. This is one plasticity zone that we have recently discovered in our group C genome. It's about a 100 kilobase region which has two tandem insertions of the same phage. Okay? And there are several different exotoxins also encoded within this region, and this region seems to be highly mobile in the sense that it also has, in addition to phage insertions, transposons 916 and the 5252 transposons, which by the way are conjugated transposons, also inserted in these genomes. This was really intriguing to me. So we have these genomes, and we have almost every strep genome that you look at tends to carry a different complement of phages. Okay? No two group A strep strains. We have 15 genomes available for that. No two group A strep strains seem to carry identical bacteriophages. Each strain, each M type seems to carry its own complement. So why does every streptococcal strain carry its unique complement of phage genes? Why can't phages move from one strep strain to another? And I'm sure that happens in the natural populations. But we were wanting to understand what was the basis of this specificity of bacteriophage infection in various strep strains. Now, when we got interested in that question, simultaneously a new discovery was made which identified these nice regions in the genome of bacteria which are called as CRISPRs. And CRISPRs basically stands for clustered repeats of clustered regions of interspersed palindromic repeats. And what these are basically a set of genes which carry this repeat sequence separated by a spacer sequence. Okay, so this is a direct repeat, and each of these direct repeats are separated by a spacer sequence, which is about 30 to 35 nucleotides in length. In all cases, the spacer sequence comes from the phage sequence itself. Okay, so the spacer is acquired from the phage sequence. And what the role of these, I won't go into the details of the dynamics of CRISPRs for one major reason, we don't know them as of now because the discovery was really real. The recent. What these CRISPR spacer ingredients do, once they're processed by these Cas genes, is sort of function in a manner which is similar to small RNA-mediated silencing. Okay? And what do they silence? What they silence is the phage RNA, which is complementary to the spacer sequence. Okay? So if identity is found, the Cas genes in complex with these direct repeats and the short RNAs flanking them will bind to the phage mRNA and lead to its degradation. In other words, providing immunity from subsequent infection to bacteria. So what we wanted to do was ask, do group A strep and group C and G strep carry these CRISPR loci in their genomes? And we obviously started with group C strep because most of the group A streps were already published without focusing on CRISPRs. And what we found were two CRISPR loci, which we call as CRISPR1 and CRISPR2. So what you see in these are the genomic maps of the CRISPR-associated gene or the Cas genes carrying these repeat spacer arrangement. Now CRISPR1, for example, carries 15 distinct spacers. All of these spacer sequences presumably identical to some phage somewhere. Okay? And CRISPR2 similarly carrying 6 spacers and 5 repeats. Now this over here just shows you that the repeats fold into a stable RNA, take on a stable RNA fold. Some of the spacer sequences in both CRISPR1 and CRISPR2, most interestingly, were identical to bacteriophages which are prevalent in group A strep genomes. Okay? Some of them, and I say some of them very guardedly because only 2 in this and 1 in this, bears complete identity to a bacteriophage present in either M type 1 or M type 3 phage. Okay, so phages in those group A strep will not be able to enter and infect this group C strep. So the next question we wanted to ask was, are these CRISPR loci that we found in our sequence genome, are these unique aberration of our strain itself, or is this CRISPR, are these CRISPRs more general and more common among other group C and G isolates? So we simply did PCRs, and we were able to show that in a sample of about 35 odd group C and G strains, genetically diverse strains, these CRISPR loci were present in virtually all of the group C-G strains that we tested. And the icing on the cake was that none of the group A strep genomes that have been sequenced to date, nearly 15 of them, seem to carry these CRISPR repeats. So these group A strep strains, during the course of their evolution, have somehow lost these CRISPR repeats, which makes them uniquely susceptible to more and more bacteriophage infections. Now, they don't carry the repeats, but they still seem to carry the ancestral remnants of the CRISPR-associated genes. So it's virtually the deletion of the repeat spacer region rather than the Cas genes themselves that has taken away the immunity from group A strep from bacteriophage infections. Which then raises the question if phages, whether group C and G streps, if all spacers, whether they have identity to group A strep phages or not, it does make transduction very tricky. Because if some phages cannot infect a group C strep strain, then they cannot be a reliable source for moving DNA from one species to another. So what are the other possibilities through which we can explain how DNA moves from one bacterial species to another? Strikingly, what we also found in our group C strep genome is this very unique type 4 secretion system. Okay? Homologs of a type 4 secretion system which are present only in group C and G strep. And group A streps do not possess this type 4 secretion system. But for those of you who may not be aware of what a type 4 secretion system is, it's essentially a battery of proteins that a bacterial cell makes and uses as an injection apparatus to translocate proteins directly from the bacterial cell into the host cell. Now, some of these type 4 secretion systems tend to be what we call as conjugators, meaning they can not only throw out stuff, they can also take in stuff, most notably DNA. Okay? So we looked at the homologies. We know that group A streps lack this type 4 secretion system. Group A strep is a naturally non-transformable species. So we tried to look, okay, so what are the closest homologs of this type 4 secretion system? And surprisingly, we found the closest homologs were perfectly conserved in their synteny in another strep called as group B strep, which is the causative agent of most neonatal meningitis in the United States. So this could provide one alternate scenario through which group C and G streps could take up DNA, floating around in their environment, and then subsequently integrate it into their genome. So right now, just summarize all this stuff. The model that we are trying to propose is sort of something like this, and this penicillin should not have been there. It's a cut and paste anomaly. I was making the slides like about 2 hours ago. So what we think is that group A strep and group C and G strep started off from common ancestor and then split as a consequence of speciation, probably driven by different ancestral ecologies, and then continued to evolve along their own individual trajectories. The easiest way to imagine different ecologies would be, let's look at a human host versus another vertebrate mammal, but not human. So during the course of the evolution of group A strep, some of the key events that we have detected include the loss of the type 4 secretion system, loss of CRISPRs, loss of several different metabolic genes, the examples of which I purposely not presented because it just makes the talk a typical genome-wide talk with lots of arrows and italics, transcriptional regulators, and the introduction of new phages bearing new toxins. Okay, so this is what what we see today. Group A streps lacking all these things but carrying new phages and new toxins. Now, a rather similar thing should have happened to group C strep. Until somewhere at this hypothetical time point, there was an ecological shift in the niche of group C and G streps, which the easiest way to look at is that wherever they were evolving earlier, they somehow got reintroduced into the human gene pool, into the human host. And at this time point, okay, with this ecological shift, what we are seeing is hardly any group C and G strain which should have followed its ancestral evolutionary trajectory. Rather, what we see, and this is putting together our genome data and our limited sequencing data from other strains, what we see is a movement of the current group C and G strain deviating from their normal projected path and moving more and more towards looking and appearing like group A strep. Okay? Now this line is solid because for this we have genome-wide data. These lines are still dotted because we're still waiting for monies from someone to help us sequence more group C and G strains so we can validate this hypothesis further. So in other words, as a consequence of this ecological shift, a proposed ecological shift, what we think is that this ecological shift is driving this asymmetric horizontal gene flow from the pathogen to the commensal, in turn making the commensal look more and more like the group A strep strains themselves. And there is clinical evidence for this because when group C and G, when they cause disease, the clinical spectrum of that disease is identical to that of group A strep, with the exception that group C and G never cause autoimmune sequelae like rheumatic fever and rheumatic heart disease. So one of the predictions of our model is that if this event are true, then the model prediction is that sooner or later there will be lineages of group C and G strep which will be more adapted to the human host environment and would probably be as capable of causing severe invasive disease as group A strep is in the very near future. So I'll just leave you over here and I'll just tell you that much of this work was not done by me, but some of the very talented undergrads that I have in my lab. People like to say with genome analysis that we have a pipeline for analyzing our data. We do have a pipeline, but these are the drivers of that pipeline. Okay? And funding support was given by a couple of people, so I'll be happy to take any questions.
What do you mean by very near future?
Like now.
It's happening now.
It's happening now. We're actually seeing epidemiological evidence where group C and G strep are causing severe disease, no longer limited to individuals who are immunosuppressed, but also in individuals who appear apparently healthy. So we're seeing a shift in our current practice.
I somewhat, why were you shocked that the metabolic genes predominate? They predominate in the genome. So if there is no selection during transfer, you could see a selection during retention. But definitely metabolic, I was more surprised that there were like tRNA genes and maybe
Yes,
ribosomal
that was
genes.
also shocked. Yeah, the reason we were shocked with metabolic genes was because being a conventional pathogenesis person, I don't think in terms of metabolism at all. I think in terms of virulence. So I'm looking for genes which are going to interact with the host. In that sense, to me, it was a little striking that the metabolic genes were moving in that dramatic fashion. Not that they wouldn't be expected to or anything like that. Yes, they do make up more of the genome.
Yes. In your final run, the model prediction, how much did it take? Time?
I don't know.
I guess.
I think so. The easiest way to look at it is I can predict this ecological shift possibly at the onset of agriculture. Possibly. It's a, just animating because that's the time we are, trying to protect animals rather than just eat them. So I think this ecological shift possibly at the time of agriculture, but going back, how much back, I don't know. I have no clue.
Is there any way that you can?
Yeah, there are algorithmic ways using evolutionary theories, but there is no fossil record. I can give you dates based upon synonymous, non-synonymous combinations, and then I'll tell you 100,000 years plus or minus 75,000. So I don't want to go there. I would rather focus on what is the consequence of this kind of movement in terms of evolving pathogenesis.
Are there any patterns with the people who are now, succumbing to these strains in novel ways? Any emerging patterns of susceptibility of those hosts at all?
They tend to be more from developing countries, where the incidence of the commensal seems to be more than actually the pathogen. So that's one major pattern that seems to be emerging.
Is there any evidence, given the question about origins in history, and I know you don't want to talk about it, but is there any way of detecting destructive damage in hard parts, in bone or anything like that?
See, the beauty of damage that occurs to the heart, for example, or the kidney, is that the bug is not in those tissues. The bug is still in the throat while it's damaging your heart. And we don't know the precise mechanisms, but we think it's an autoimmune response. It's not a direct toxicity response that the bug has to be in the tissue side to cause the damage. So, no. Yes, we-
I realize this is different from the kind of system you're looking at, but it raises some interesting questions about whether people are, if you've got this huge horizontal gene flow, particularly if for example, you have the same host, and one was commensal and one wasn't, can you target possible things in the commensal that might be particularly dangerous in the future if they were to move to the...
That's one of our ultimate goals, to identify and catalog these species-specific genes and screen human sera for antibody responses. And one of the major problems of why in developing countries where the incidence of both commensal and pathogen tends to be high is one of the reasons if we're not focused on these is that the tools that we use for diagnosing group A strep, the antibody-based assays, those proteins are same in the commensal. So when we do antibody response assays to say, let's say, rheumatic fever or rheumatic heart disease, we actually don't know whether we are looking at group A strep or group C strep. So one of the first idea is to look for those species-specific genes which can allow us in a targeted manner to conduct proper epidemiological studies, where we can identify with confidence whether the antibody response that we are catching is because of the commensal or because of that. And then in a reverse manner, in the long term, hopefully find a target that we can use to treat one or both if required. All right, so
Okay.
sorry for keeping you for dinner.
Now, so thank you, Awdhesh, very much, Mr. Pakistan. We have dinner, which will be out there any minute now.